Yeah, hello everyone. I am Sami Ullah. I'm working as a scientist in the tumor biology group at the German Collection of Cell Cultures and Microorganisms in Germany. In short, it is called the DSMZ, and we are actually one of the Europe's largest cell line repository, and we not only keep the cell lines that are submitted to us, but also we develop cell lines by itself, and we try to actually also do the quality assurance, and we offer sales and services for different kinds of cell lines. If you have any questions during my presentation, or if you want to connect with me, or if you anyways have any other questions regarding cell lines development process or about DSMZ, you can contact me on samibiotech@gmail.com. The email address is in the first slide of this presentation. So what I will be talking to you about today is about optimizing the cell lines development process, and that would be especially in the context of bioprocessing. And in bioprocessing, it will be in the upstream bioprocessing. So to develop the cell line by itself, and then of course we would go to the upscale, and then from there onwards, how to develop them into the industry. So with that, I would like to start the presentation. The first thing that I want to talk about is that why would we need a cell line for bioprocessing? And that comes with the term biologics or biopharmaceuticals mostly. And the term biologics or biopharmaceuticals are almost similar to each other. It only refers to a really broader range of products, especially the medicinal products. All of them actually share some kind of common features. Now we know that in the traditional medicines, we synthesize compounds or we synthesize medicines in the laboratory, and mostly it is kind of a chemical synthesis of the compounds, so synthetic chemistry is involved there. But in terms of biologics, the bioproduct are in short, the product itself is synthesized in a living cell. That is why it is called the biologic then. And in the pharma industry currently, a lot of drugs are being produced, which would be termed biologics. If I give you a short example, that would be from, for example, the 2014 to 2018. So in these 4 years only, about 68 monoclonal antibodies, 23 hormones, 16 clotting factors, 9 enzymes, and 7 vaccines have been produced using a biological system, which means that they have used cell lines to develop such kind of products. And these have been already approved or are in the process of approval right now. So before going further into the process of how these cell lines are developed, I would like to mention here that what kind of systems can be used for this process. So the objective here is that to produce a recombinant protein, or for example, a monoclonal antibody, or any other kind of biological material. Now, we can use different kind of systems, and mostly these systems are called the expression systems because these proteins would be expressed in these systems. So for example, bacteria, and that I have given an example here of E. coli is one of the most common examples of a bacteria which is used for production of different kind of materials. Similar way, yeast can also be used, and in that case, we have Saccharomyces, one of the examples. Insect cells, such as SF21, these can also be used, and my focus here would be on mammalian cells, and here the best example is CHO cells, or the CHO cells, which are the Chinese hamster ovary cell line. Most of these cell lines are most of these biological systems that we use, so we have to select them based on our product itself, based on what we actually want to produce at the end. So one example would be that if we want to produce, for example, the viral particles, so for that, the best system here would be the insect cells, the SF21 cells, for example. The mammalian cells are the CHO cells. These are actually selected if we want to produce certain kind of vaccines, monoclonal antibodies, recombinant proteins, or hormones as well, for example. So for such kind of system, then we would use mostly the mammalian cell line. Now, to give you some insights into the mammalian cell culture and how they are used and why they are used for biologics production. So mostly, as I said before as well, they are used for the vaccines, antibodies, or recombinant proteins production. But why exactly are they used is because of the biological system itself. As these recombinant proteins would mostly be used by a mammal itself, for example, a human being, these proteins would probably not need any further modification. And that we can ensure if we use a system which would also ensure the post-translational modification, which is a necessary part of these proteins production or proteins manufacturing. In other words, protein folding as well is one of the steps which is required for the protein production, because usually we have the amino acid sequence which is produced by a gene. So that would be folded into a protein as well, and further the 3D structure of this protein would be made within the cell. So that is why we would use then mammalian cell culture for that. So we can actually use such kind of biologically available phenomenon and use these phenomenon to produce our protein in question. Now, this is actually the upstream process, as I said before as well, and it will have a different kind of steps involved in it. So the most important here is that we will transfect our gene of interest, which would encode for a specific protein, into the host cell. And for that, we can use different kind of vectors, for example, or plasmids. And then afterwards, we would select the clones of cell lines, which would have, for example, high productivity, a high proliferation rate, even in terms of the cell itself, it should be having the characteristic of the cell. That was actually what we begin with. And then after that, we will subsequently go for the scale-up process. And the scale-up process is that once we are producing them in a small Petri plate or in 96-well plate or 24-well plate, and afterwards we would go to the flask, and ultimately we will go to the bioreactors. And in each step, we should be ensuring the quality of these clones of cell lines as well. So the different types of these mammalian cell cultures which are used, I have tried to put here some of them. So depending on the purpose, as I said before, we would use specific cell types. For example, if we want to produce proteins or antibodies or vaccines or even complex therapeutics, so for that we would mostly use the CHO cell line or the BHK, HEC293, NS0, or even the insect cells. If we want to produce viruses for gene therapy, for example, then as I said before, mostly we would use the insect cell lines, and this, in this case, is actually the vivo cell line. We can also use HEC293 cells, which are one of the most powerful cell lines available, which can be used for different kind of purposes. We can also use C61 cells. And then finally, if we want to go for the cell therapy or regenerative medicine or even personalized medicine, so for that purpose, we mostly use pluripotent stem cells. And these are mostly embryonic stem cells or adult stem cells. So these would be then differentiated into specific cell types, for example, and those cell types would be then given for cell therapy or regenerative medicine. These are only the examples, and there are many of other cell lines available. Mostly these cell lines would be still modified in terms of expression of specific gene. And then afterwards, we would be actually giving them or using them actually for our purpose in question. So the process of a cell line development, that would be for us the initiation of the biologic production, is I have summarized them into 5 steps. And these 5 steps are, for example, the transfection. Once we transfected the cells, then we would select the cells which are really transfected, which do have our gene in question, or the gene of interest. So that would be actually a pool of cell lines, a pool of cells of that particular cell line. And that would be then, we would submit them for pool selection. So we would provide some kind of pressure to these pool of cells, and then we would select the cells which do have our gene of interest. So once this pool is selected, it's still a pool, so we do not know that how many copy numbers are available in these cells, and in which cells how much copies are available. So that is why we will then go for the single cell isolation or single cell selection. And once we have isolated the single cells, we would produce the colonies of these single cells, and then we would assess each colony for different kind of quality parameters. So once we have isolated the single cell and produced a colony of that single cell, which is for our purpose in question is working the best way, then we will expand that particular cell clone and then characterize it as well. And characterization, of course, is dependent on different kind of vectors. So once we have that thing as well, the final step would be the optimization process. So optimization would be not only for the cell line as well, but also for the biologic production part as well. And once we have optimized this process, we would scale it up. Now, one thing that I would like to actually mention here as the cell banking part. And my advice to you is that cell banking should be done at each and every step. So once we have transfected the cell, we have a pool of cells available. So the ideal way is to actually pool, this pool should be stored in some way. Mostly these are stored in -80 refrigerator or the deep freezer. And then if we have clones available, so those clones also should be stored in the same way. After characterization, after optimization, at each step, it is better to bank the cells somehow. And either, for example, -80 fridge or also in liquid nitrogen. Why is it so much important? Is that if we do that, if we have any problem in the future steps, for example, if we do cell banking in step number 2, but we have some problem in step number 4, we have some contamination, for example. So in that case, we can again go to step number 2, check out the cells from our cell bank, and then continue from step number 2 and go forward to 3, 4, and 5. But if we do not have those cells available in our cell bank, of course, we should be going then back to the transfection part. And that, again, is a lot of labor, a lot of cost, and a lot of time, which we would be needing for the whole process. So I will be presenting each and every step here, how we do it at DSMZ, or how we do it in general for the cell line development, and how I have done it so far. So the first step here is the transfection part, and transfection is a really straightforward thing. We have a vector. So in this case, I'm giving you an example of a plasmid. Now the plasmid has some certain restriction sites. It has other parts as well, but it has some restriction sites. We will be taking in consideration that these restriction sites should be matching our the restriction sites in our gene of interest. So the flanking region of these gene of interest should have some restriction sites, which would be matching the same sites at the plasmid. So when we apply some restriction enzymes to these plasmids, it will cut the plasmid at those sites. And once those are cut, it will be hollow. And then we would ligate our gene of interest using ligases. And then we would be having our plasmid containing our gene of interest. Now, such kind of plasmid would be really available for our purpose. That would be the cell line development for that specific kind of gene or protein. So how exactly it is done in the lab, or how we do it in the lab, is that we have certain specific transfection reagents. Different companies are providing different kind of transfection reagents. We mostly do it with lipofectamine, for example, and this lipofectamine reagent, transfection reagent, is first of all suspended in serum-free media. Then we have our nucleic acid, which would be DNA, for example, coated with certain specific restrictions enzymes. Now we would mix them up, incubate them for a certain time, and then add them to the cells, the whole mixture itself, and incubate the cells with the transfection reagent containing our nucleic acid. Now the important thing here to consider is the confluency of the cells. Of course, the cell line itself, which you are using, should be selected based on your purpose, but the confluency is also important. The best way is to actually add these transfection reagents containing our nucleic acid when the cells are 70 to 80% confluent. In most cases, if it is 80% confluent, it is better. Because we are adding incubating the cells with these reagents for about 2 to 3 days, which means that the cells would go their normal proliferation as well. That is why certain space should be available for our cells to grow. And that is why we actually keep the confluency at 80%. The lower confluency should also be avoided, because in that case, we will have a lot of surface area available for the transfection reagent, but no cells would be available on the plate. So that is why we will go for the 80% confluency in normal cases. The second step in the cell line development process is the pool selection. Now you have already transfected the cells. You have a pool of cells. Some of them should have 1 plasmid, some of them should have 3 or 4 plasmids, and some of them should have no plasmids at all. So now to select the cell which actually has the plasmid in question, we use different kind of assays or different kind of strategies. And this, for example, I will give you example of antibiotic resistance or fluorescence related gene. So normally in the plasmid, we will have an antibiotic resistance gene, for example, for ampicillin, streptomycin, or gentamicin. We can also incorporate the fluorescence gene, for example, the RFP or the GFP, it should be either red or green in color, to the plasmid itself when we are incorporating our, of course, the plasmids can come with the specific fluorescence gene or with a specific antibiotic resistance gene. Now, when we have our pool of cells, we would grow them for the antibiotic resistance genes in a media, which will have that antibiotic in question. So now when the pool of the cells which do not have the plasmid inside them, they would die in that media because obviously they are not antibiotic resistant. But the pool of the cells which have the plasmid inside them, they would grow on these media. The same way for the RFP and GFP, we can use microscopy, or we can also use FACS sorting of the cells. Then again, once we have the selected pool, we would isolate the single cells. And for the single cells, we are using different kind of strategies. These different kind of strategies can be, for example, that we can use the manual picking of the single cells using a microscope, so with a spatula. We can also sort the cells by using fax machine, or we can use automated single cell isolation machines as well, which are nowadays available and are given by different kinds of companies. This is one of the most important parts of the cell line development because you want to select a clone which actually would be the most beneficial for your purpose. So that is why a lot of care should be taken at this step. For the manual selection, as I said before, you have a microscope, and under the microscope, you are manually picking up the cells. So this I have done, for example, during my PhD time in 2015, but nowadays different other methods are available, so I'm using those in my research currently. So the manual selection, of course, it should be done under the laminar airflow hood, and as it is shown here, you have the cells under the microscope in a laminar airflow hood, and then what you do is you scrap the cells. But before that, you suspend the cells in really high volume of media, and then actually mix them or plate them in a Petri plate. So you have a lot of surface area available for the cells. So for example, it should be 10% or 15% confluent, so you can actually pick up single cells there. And that is usually done with a pipette tip, using a pipette, and you just directly scrape the cell a little bit, and then afterwards pick it up using the pipette. And once you have that, you transfer it to the 96-well plate, put the corresponding media on top of it, and then let it incubate so it can proliferate. So care should be taken here because it is a manual process that you are actually picking up only single cells. The second way to do this single cell isolation is by fax sorting. And again, the fax is a normal machine which has a copet and some nozzle as well. And with a droplet, you actually are taking out only single cells through the nozzle. And each droplet should be containing only a single cell. And that single cell then is directly poured down in a 96-well plate. Once you have that, then you go on and add some media on top of it and incubate the cells again. Now, currently available, the most advanced method actually is the automated single cell selection. And here I'm giving you one example from one company where they are using this automated machine to pick out single cells. It is actually having dual system. And the dual system actually is that, first of all, it is using kind of like the fax sorting. But in the fax sorting, we are giving a lot of pressure to the cells. Here, in this case, they are not putting that much pressure on the cells. So basically, it is kind of like the droplet method, and the pressure applied is only through squeezing the cells in a droplet. So no pressure is applied on the cell, and that's why you will have a lot of high viable cells. So then again, it is using and ensuring the quality of the single cell isolation process as well. So it is using the microscopic methods, and that is that one of the camera is actually on the nozzle itself, and one of the camera is directed on the well of 96-well plate. So it ensures, first of all, that a single cell is actually getting out of the nozzle, and then it ensures again in the plate that there is only single cell which is poured down in the 96-well plate. Now I'm talking about this automated machine because usually with the picking up manually the single cells, you are applying a lot of pressure. And that pressure cannot be good for the cell as well because it is stress, it's mechanical pressure. So you normally have less viable cells, or you can actually alter the cell properties as well. The same way is with the fax sorting, it is again a lot of pressure. And with the automated single cell isolation, then such kind of pressures are avoided. Cost is avoided, labor is avoided, and time taken for single cell isolation is also really low. If I give you one example, when I was using the manual single cell isolation, I actually ended up taking days for picking up colonies. But in case of automated, it can actually take a few hours. So at the end, you have single cells. You plate them in 96 well plate. You put proper media on top of it, and then let it grow. And at the end, you have colony. Now, those colonies are then screened for the high producers, which will produce the high amount of your protein in question. And then that high producer can be tested using different methods, for example, PEX or ELISA, if you want to test for antibodies, or if you're producing antibodies. Then the next step in this process is the expansion and characterization, which usually is done by live cell imaging to test the proliferation, which cells are proliferating faster, which are slower as compared to the original cell line. Then you do SDR profiling using PCR to confirm that it is the same cell type. You also do microplasma testing for possible contamination. You check for the cell surface markers as well to further ensure the cell type. And then you expand them in larger plates and flasks. You biobank them, and then you actually test them for high producers as well. Now, the media selection process, as I said before, it should be the serum-free media. And the serum-free media is essential because usually in the serum, it is batch-to-batch variation between each serum which we get. There's a possible chance of contamination within the serum itself. And usually the cellular growth and physiological responses of the cells is inconsistent in different kinds of serum. And not to forget here is also that certain compounds are sequestered in the serum itself. So different kinds of alternatives are available for the FBS media, and those can be, for example, the serum-free media. So in general, they do not only have serum. The xeno-free media, so it is not taken from the animal source. Animal component-free media, even protein-free media, and finally the chemically defined media. I prefer the chemically defined one because in that case, you know exactly that what is inside your media and how to further identify that, okay, what kind of points I should be taking in consideration before I go for the production of specific biologic. Then the final step is actually the scale up or the optimization of our cell line that we have produced. And that we can go back to our initial stage, which is that we have produced, for example, our cell line in 96 well plate, then we transfer them to 48 well plate, 24 well plate, and finally to the small flask, which is 25 meters square, and it takes like 5 to 7 milliliter of media. Then we go to larger flask and even larger and even larger. And in each passage and in each flask that we are expanding our cells in, we are checking for the proliferation of the cell, we are checking for the viability of the cells, we are checking for the cellular properties itself, and finally we are checking for how much of our biologic in question is produced at each step. So that actually ensures that whatever clone we have picked up in the beginning, it is the clone which has the potential to go to the industry, to the bioreactors, and to produce our biologic in question. And that is especially important then, because you are not going on with only a single clone, but you actually pick up 5 to 6 or 7 clones, which are the most promising clones. So at the end, if there is anything happening, which would happen in most of the cases anyways, then you have the clone, the opportunity to actually pick up another clone, which would be the best one. So to finally sum it up, what I talked to you about is that the mammalian cell culture is a robust and an accurate method to produce your biologics. And the cell line selection process is dependent on the product itself, of course. And some of the steps in cell line development can be automated, as I said, for example, in case of the automated single cell isolation method. And in that case, you actually reduce the time consumed during the process and also reduce the cost. Serum-free media, I showed you that it is one of the most promising methods to actually produce your biologics in it. And that is in any way the best choice for cell line development as well, because you should be taking in consideration that the ethical reasons for the FBS use as well. FBS is anyways produced from the fetal calf serum, which are actually the live animals in third trimester of pregnancy. So yeah, ethically, it is not a good idea to use the FBS in such a large amount. And finally, I talked to you about the cell banking as well, that it is a good idea to bank your cells at each step, because that way you will not only reduce your cost, but also the labor involved in this whole process of cell line development. And with that, I would like to thank you for listening to my talk. And if you have any questions, as I said before as well, you can contact me directly on my email address, samibiotech@gmail.com. If you want to network, if you want to get more information about the cell line development or about DSMZ, you are free to contact me. And with that, thank you so much. And I'm looking forward to the questions in the chat box.